So today we have Dr. Ronald Jenner, who's going to be giving our Halloween-themed lecture on the frightful tales from the world of venom. So Dr. Ronald Jenner is a research leader in the Department of Life Sciences at the Natural History Museum, where he works on neglected venomous invertebrates and the conceptual history of phylogenetics. He was the scientific lead for the Natural History Museum exhibition Venom, Killer and Cure, which was really, I went and got my jumper. It's a slow Loris. So let's welcome Dr. Ronald Jenner.
Thank you. Can everybody hear me in the back? Excellent, thanks for the invitation. I see there are some younger members of the audience. I will show some photographs that are at least a bit upsetting, because there will be snakebite photographs. There will also be genital photographs. So I just give you a heads up. If I remember, it's only 2 or 3 slides. But I thought I'd probably have to tell you this. Right, so frightful tales from the world of venom, and then I have a cute little frog. So if that seems incongruous to you, just be patient. We will get to it. It's nice to be in the Linnean Society. I mean, I've been a systematic biologist since the late 1990s when I started my PhD in systematic biology. But I'm not here as a systematic biologist. Weirdly, I'm here to talk about venom in the Linnean Society, which is odd. But perhaps as a way of introducing why do I work on the venom, and why am I no longer a systematic biologist, although I think I am. I will actually also set my own alarm, because I do tend to talk over time. But you gave me a bonus minute. Awesome. It's not even 3:30 strictly. There we go. Let's have a look at what the average working career of a systematic biologist might be and that of a venom researcher. Well, let's take, for instance, somebody who's doing extremely important work for the International Code for Zoological Nomenclature. This is Doug Uniga from the University of California, Riverside. He's an entomologist, a taxonomist. And Doug does a lot of things, including field work in such exciting places as rain puddles on roadsides. Doug, no matter what he does, whether he sits behind a microscope, shows specimens, he has a very even temper. This is not a man who looks like he is extremely enthusiastic, or if he is, he might hide that quite well. Of course, as befitting a true scholar, he has thick glasses, whoops, and he has t-shirts that we all like to wear as biologists with animals and plants on them. This is very different when you look at the representative venom researcher. This is Brian Fry, known for his work on reptile venom biology and evolution. Brian does field work also, but he does it in more exotic and exciting locations. For instance, he will follow Komodo dragons and prod them with a stick. Afterwards, they kiss and make up. He also wears glasses, but only really as a fashion accessory, and his shirt comes off all the time. I've been with this man at conferences like here. He basically shows his tattoos. This is what he does. And he has quite a few. He has the skulls of Komodo dragons on his back, a snake. And this little thing here is the chemical structure of adrenaline. It gives you a hint of what some venom researchers are like. I hasten to add, this was not the reason why I went into the field. I liked his work rather than his perhaps slightly narcissistic tendencies. But one thing that struck me immediately when I started to shift from working on, say, crustacean phylogeny, which is flatline boring, to working on venomous invertebrates, is the media cares, really. I suddenly found myself on Channel 4 talking about my Venom book and about the Venom exhibition that Leanne mentioned that ran in the museum last year. Suddenly, there were things on tweet that were very kind to me. Suddenly, people actually took interest in our work, such as on the venomous remipede crustaceans, where on a science blog they said they were very excited that we discovered the world's first poisonous crab in Western Australia. I put this on my CV, but this is not a crab, it's not poisonous, it's not from Western Australia. You get the drift? You know, science reporting not vetted by peer review can be not so good. This is actually a big problem. What does poisonous and venomous actually mean? Well, let me illustrate this with 2 frogs. And here we have on the left Dr. Valerie Clark. She works on poisonous frogs, such as this golden Montella frog from Madagascar. And she's interested in the compounds they use for defensive purposes. So what you do is you crawl through the undergrowth, you pick a frog up, you lick it. If it's tasty, then you throw it back. If it's not tasty, it means it probably has defensive compounds. And you might bring it to a real scientist like a chemist to figure out what these things are. So poison is something that is basically delivered passively by absorption through the skin, by swallowing. Some poisons can be inhaled, so no special mechanisms involved. It's also typically non-protein. So, for instance, these frogs have alkaloids. Think nicotine, think cocaine, things like that. These are simple organic molecules. Often, the toxins in poisonous animals are exogenous, which means, like these frogs, they get them from the toxic food they eat. They don't make those toxins themselves. And by and large, they're used for defense only. Venom, very different. Venomous creatures have special delivery mechanisms to inject venom through wounds. So you're thinking claws, spines, fangs, like in snakes. You're thinking about complex cocktails of molecules, the most important of which are proteins. The animals make them themselves. They have their genes make these proteins, and this is what comprises the venom, and they're used for a host of biological roles. Now you're thinking, wait, this is a frog. Is that venomous? This is a venomous frog. The slightly apprehensively looking scientist here is Carl Jarrett. He once picked one of these up a couple of years ago, and that was a bad mistake, because if you look at its skull has all these pointy bits, especially in the snout area. Covering these pointy bits is a skin full of toxin glands. When you pick up the frog, it headbutts you. It's very cute. It's and it rubs its head on you. That causes immediate radiating intense pain that lasts for hours. Carl Jarrett did not like that. Carl Jarrett wrote a Nature paper on this. So this is what venomous creatures do. And it became even more interesting when they started looking at how toxic are these little frogs anyway. If you compare it to, say, a Brazilian pit viper in the genus Bothrops, because these are Brazilian little frogs. And if you compare it to a snake like this, which can cause wounds like this, the frog's venom, gram for gram, is 25 times more toxic. Thank God they are tiny. So, but the moral of the story is even such innocuous little creatures like frogs can be quite dangerous. Now, people still don't get it. Fenneman Poison, we were doing the exhibition, we were doing the book. And as we finished the book, or we're wrapping it up, they said, we've made you an awesome cover. I said, it's wonderful. You know, it's warning colors. Yellow and black in nature means bad, right? Wasps, bad. Don't touch yellow and black stuff. But this is a poison dart frog skin. Cannot really sell a Venom book with a poisonous creature on the cover. Doesn't really work. Oh, we go back to the drawing board. They come up with this wonderful yellow and black python. Pythons are not venomous. They are constricting snakes. So I said, you can't really do this. We put so much effort into that. Said, yeah, right. So eventually they came up with a black and yellow banded crate, which is a member of the Cobra family. Very dangerous, very venomous. So it ended well. Now, venoms, when you think, most people think spiders, scorpions, snakes, and then there's a precipitous drop. It's like, what else? Well, a lot. 200,000 living species are venomous. Venoms have evolved 80 different times independently. This big tree of the animal kingdom shows a few of these instances in the colored lines. Red means predatory venoms, blue means defensive venoms, and they've evolved in the arthropods, in the snails and the worms, in sea urchins and sea stars, and in the vertebrates as well. I will just show you one slide with the creatures I very much love. They are distributed in this tree. My colleagues and I work on venomous invertebrates mostly, because most people don't study them, because they're not medically relevant. They're just interesting to comparative biologists. So I work on venomous crustaceans, pseudoscorpions, which are cute. They're 2 or 3 millimeters long, and they inject venom with their little claws, which is, there's more of those venomous species than there are venomous scorpions. I work on a thing called bloodworms, which are these ugly things, centipedes, and I recently started working on a fish because invertebrate biologists have an inferiority complex, and we also like to work on stuff with backbones, because those are real animals. So we work on a defensively venomous fish. One thing in all that diversity that venoms have in common is they are, by and large, complex cocktails. Most important components in both abundance and function are proteins and peptides. Think egg white, that is a type of protein. Peptides are simply small ones. How do we know they're complex cocktails? Well, take Conus geographus, the geography cone, a marine snail that is pretty ambitious because it likes to hunt fish, which is a big thing for a cone to do, and I will explain later how they do it. If you separate its venom in different components, and then you inject those venom components in mice, and you score what they do, you get a very, very interesting spread of behaviors. You get the mice that run around in circles, you get the head swingers, you get the twisted jumpers, you get the sleeper climbers, whatever phenotype that is. They fall asleep, they get up and must climb, fall back asleep. You get worse things, you get convulsing, bleedy, die mice. What happens is, if you get stung by a snail like this, the geography cone, this size, you die within 12 minutes to 10 hours because it paralyzes you completely. It's very painful, but it's full of neurotoxins that wipe out your nervous system. And this is the way, of course, in which a snail can hunt a fish and also defend itself against large predators like ourselves. However, it gets weirder. It's not just that we live in a world where snails can be dangerous. Cute little things, like on your shirt, Leanne. You get these cute little primates, the slow loris. This is its defensive posture. It's like, whoa. This is Brian Fry again. He's holding this primate with a rubber glove. Why does he do that? Well, if you don't, you may be in trouble. This is George Madonna. I say is, I don't say was. It's still fine with him. He was in Borneo, saw a slow loris in a mango tree, this specific specimen, thought this is so cute. He climbed into the tree, they both fell out, picked up the animal, bit him in his finger, and it made him very sad. And it was bleeding. And then an hour later, anaphylaxis was setting in, and he started to look like a cartoon character. Slow lorises have already a confirmed kill rate of 1, in which a pet owner had 2, they were fighting, and he came in between them. Don't do that. Don't do that with dogs. Don't do that with slow lorises. And so he had to go to hospital and get treated for anaphylactic shock, which is essentially an overreaction of your body to a foreign substance. Well, that's clear. Is that what these animals use it for? Well, they use it for defense, but they use it for other purposes as well. So the interesting thing is their venom doesn't come from glands in the mouth. Their venom comes from a gland on the inside of their elbow. They lick it, and they mix the secretion with saliva. It activates it. What does that do? It causes painful, festering necrotic wounds, and they use it to fight each other for territory, for mates. So this is a healthy one. This is when he had fought, and that's what a couple of months later was he sort of recovered. If you get bitten, you can get these same necrotic, festering, long-lasting wounds as well. But there was an interesting other aspect to the story as well. These are not just venoms that are used for defense. Babies are anointed with these gland secretions, and that deters nose-oriented, olfactory-oriented predators. You take a cotton swab of this venom, you mix it with loris saliva, you offer it to a tiger. The tiger goes, oh man, that smells bad. You offer it to a bear, same story. Leopard, same story. Civet cat, same story. Then you offer it to an orangutan. They are not nose-oriented hunters, but they just all eat the cotton swab. And in fact, they have been seen to feast on slow lorises in the forest because they're small. They just slap them off the branch, climb down at the tree, and eat them. It's bad. They also use these secretions to deter parasites because if you add, for instance, a tiny bit of mixture from a cotton swab with this venom to a leech, it dies within a minute. So this is an interesting multifunctional toxic mix. So what causes the anaphylactic shock? Turns out that the component that does it is a homolog, is an evolutionary relative of what makes you allergic to cats. It's a protein called FeLD1. It's in cat urine, in cat saliva. If you're allergic to cats, stay away from slow lorises, is my advice. But it's not really that anaphylactic shock shows you the power of venom, right? Because it's your own body overreacting. The venom doesn't really do much. It's an inert protein, this FeLD1. There's 2 physiological Achilles heels that venoms attack. Cardiovascular system, which means your blood vessel system, and your nervous system. So let's have a look at these in turn. And I can always come back and illustrate everything about venoms with Brian Fry. This is Brian Fry with his PhD student, Jordan, milking a snake. For some reason, I find this such a bizarre photo. This is not how people dress in labs, but hey. And this is an incident in which the milking went well. When Brian was a PhD student, he found this member of the Cobra family in Australia, tried to milk it, it bit him. He immediately got a headache, lost consciousness within minutes, then woke up and he started to bleed badly. He started to bleed from his mouth. He didn't like that. He started to bleed from his nostril. He didn't like that. He started to bleed from his tear ducts, which he did say he thought was cool because he looked like a vampire. When he started to bleed from his derriere, he thought this is not longer cool. So for 18 hours, they had to treat him for bleeding until his blood could clot again. What had happened? Well, he lost his consciousness because the venom of this snake is full of a hormone that we have as well, which regulates blood pressure.
If you inject a lot of that into somebody, their blood pressure plummets and you lose consciousness. And this is an element of Venom Evolution. Venoms recruit normal proteins with normal roles into the venom and weaponize them, make them weapons. The bleeding was caused because the snake, Stephen-banded snake, has evolved a blood clotting factor, which itself the snake uses. You know, if it gets a wound, it needs a clot. Put it in its venom and expresses it at a high level and causes massive blood clotting. And that means that when in a short time, all your blood clotting factors are consumed and you have unclottable blood. Hence, you start bleeding internally, externally. It can get even worse because there's some other Australian members of the Cobra family, like brown snakes. And Taipans, they have recruited a second clotting factor, making their blood clotting activity 300,000 times stronger, which means as soon as it gets injected in a mouse, all these clots form, and these clots can, of course, cause strokes when they block blood vessels. In fact, if you inject such Taipan venom in a mouse, and you look at, for instance, lung tissue, you see that large blood vessels and small blood vessels are completely solid with blood clots. That's not good for you. Well, why is that? Well, Taipans are mammal specialists. Taipans eat things like this. Bandicoots. These creatures, the quolls, they can bite. They can actually kill the snake. So the snake needs to have a weapon, which means it works immediately. When a taipan bites a mouse, before the fangs are pretty much out of the mouse is dead because of blood clotting. If it only gets a little bite in, they have very strong paralytic toxins, and the mouse crawls away a meter and is dead too. So this is one of the reasons why they need such strong venoms. They want to incapacitate a prey that can hurt them immediately. Now, the second, this is a very confusing graph, but I will give you two things to notice. This is just a blood vessel, and this is all the blood clotting factors that make a blood clot. All these different targets in this system are targeted by venoms of different creatures. The one thing I wanted to illustrate here, that it's exactly what snakes target, that ticks target, and kissing bugs target, and mosquitoes target, not to kill you, but they want to manipulate your blood flow so they can blood feed on you. So venomous creatures are also mosquitoes, are also leeches, are also ticks. They use the same molecular weapons to manipulate you, not to kill you, but so they can feast on your blood. And that makes the world more scary. The second Achilles heel in your physiology, if you want to deter a predator quick or subdue a prey quick, is your nervous system. This is a block from a person in Southeast Asia, where there is a lot of snakes. There are harmless snakes, like this common wolf snake, and things that look like it, like a common crate. Common crate is Cobra family. Highly venomous, paralytic venoms. How do you tell them apart? Well, they look a bit different, but of course they're black-white banded. But he writes this, you know, is it difficult to separate? No, it's not, you know, just open its mouth and check the fangs. That is unbelievably ill-advised. Do not do it like this, not even when they're dead, because unless it has decomposed, you may still prick yourself and the end can be near. For instance, this is the late Joe Levinsky. September 2001, he was a herpetologist at the California Academy of Sciences, did a lot of work on reptiles and amphibians in Southeast Asia. He was in Sarawak, and somebody handed him a bag with what they said was a wolf snake. He stuck his hand in the bag, he pulled his hand out, and I have to quote him verbatim when he said, "That's a fucking crate," by which he meant, this is a venomous snake, this is not a good thing. This was 7 in the morning. This is the snake in question. It was only this long, pencil thin. Didn't even see a bite on his finger. He's like, yeah, gonna go and have some breakfast. Hour later, tiny little numb feeling started in his finger. So they dispatched runners to the nearest village with a radio. They came back at 3 p.m. in the afternoon. By that time, he was completely paralyzed. These are some photos taken by his entomologist friend. He had to be given mouth-to-mouth. Long story short, they had to give him mouth-to-mouth for 26 hours, followed by 3 hours of CPR before he passed away, because the weather was too bad to dispatch a helicopter to help him. Until the end, he was a very humorous man. He was conscious, fully conscious, just completely paralyzed. And he could still wiggle his toe for the first 12 hours or so to say yes or no. When some of his female colleagues, who were the only ones giving him mouth-to-mouth, said, we are getting a bit tired of this Joe, can the dudes take over? He said, no. No, he did not want that. Poor Joe died on the 12th of September 2001 of snake bite. And the thing that probably killed him is the major paralytic neurotoxin of this snake. This is a nerve, this is a muscle, blocks the transmission of nerve impulses to muscle. So you go floppy, you go flaccid. It's the same target that curare hits when you use that plant poison to hunt for, say, monkeys. And you can see, as with the blood vascular system, targets in this system of nerves and muscles are targeted by all kinds of venomous creatures, because this is the Achilles heel with which you can take out predators and you can subdue prey. Now, Joe got a little bite. It was completely painless. Kraits often come into people's houses, bite you while you're asleep. You don't wake up. It's completely painless, but you wake up in the morning unable to open your eyes. And so you need antivenom quick to help that. Some venoms are not like that, soft. Some venoms are really bad. How do we know? Well, we can do animal experiments, and those are sometimes necessary. Just take an almost random example from an issue of the journal Toxicon in the year I was born. And they injected sea wasp, which is a cubazone or box jellyfish venom, into rabbits. They remained passive. The breeding became deep and labored. Within a couple of minutes, they started falling down, kicking their leg. The animal emitted a shrill cry. 1 minute later, they were dead. Much the same story if you do it in sheep, but then with the dispassionate prose of a scientist. There was no focalization. So the sheep, in contrast to the rabbits, died silently. I'm not going to give you the description of what happened to monkeys. But what does happen if you get stung by one of these jellyfish and you get enough of the tentacles with the stinging cells on you, you get big problems. Why? This is not just a venom that targets your blood vascular system or your nervous system. Cubazone, box jellyfish venom, targets absolutely everything. It destroys your muscles, your nerves. It poisons your blood, it destroys your cells, it is toxic to your heart, and it attacks your skin. It's full of what we call pore-forming toxins. They insert themselves into cells and they go, and the cells pop. If you start popping cells and your tissue becomes destroyed, you get big problems. You get very big problems indeed if you swim into one, such as poor 10-year-old Rachel Shardlow did. This is one of her legs. She got tentacles on 30% of her body. She went into cardiac arrest immediately, was in a coma for 3 days, but happy news, with medical care and antivenom and some skin grafts, she's now a happy girl and she's fine. But this goes to show this is a jellyfish that uses its venom to catch fish. You need to have an all-out fast weapon if your maximum speed is about this and you try to hunt prey that goes so there's a reason for this. But not all hunters do it so dramatically. Go back to the geography cone. The geography cone, when it stings you, is using its defensive venom. It has 2 different venoms, one for defense, which is like a bazooka, and then a sniper gun for when it hunts little fish. And it starts its hunting with the most bizarre techniques. It swims up, or crawls up rather, to a fish, and it opens its rostrum, its mouth, and it wafts what seems to be secretions to the fish. The fish doesn't swim away. And people have discovered that this snail has evolved an insulin, which is a hormone that all vertebrates use to regulate blood sugar, and it gives the fish instantaneous hypoglycemic or diabetic shock, because these peptides are absorbed through the gills. The fish doesn't want to swim away because it's like, oh, this is awesome. I feel so hypo. And then the snail can actually envelop the fish entirely, even schools of them. Then when they're inside that big trunk, it shoots a venomous harpoon to finish them off. So this is one of the ways, and this is just to show that the insulin protein of the snail looks very much like that of fish, in which these creatures can actually envenomate you from a distance in a very refined fashion. Some creatures do it with the other side of their body. These are acrobat ants, and they are very cute. They hunt by putting their derriere up, presenting a droplet of venom which is so volatile, it evaporates and it's toxic, and anything with a thin cuticle, like these poor termites, will essentially be farted to death. And so they close in with their little butts, closer and closer and closer, and then drag it away. If they're confronted with big enemy ants, they do the same thing. So here's a nice photograph where you can see it present this little droplet of venom. So they basically use a wonderful no-contact venom to get what they want. Of course, insects are magnificent if you want to understand venoms, because venoms in insects are not just used for predation. They're used to feed the babies. A lot of parasitoid wasps inject their venom to manipulate hosts to create meat ladders for the babies. They use their venom to keep the nests free of microorganisms and pathogens. They use their venoms, for instance, even as alarm pheromones or sex pheromones. We have female wasps using the perfume of their venom to attract males to induce them to mate. But of course, most of what insect venoms are known for is painful stings, especially in the social insects, especially in ants and bees and wasps. Why? Well, these have a very important resource to defend. They have nests full of honey, full of, if you're a honeybee or a honey wasp, full of babies, full of eggs. You need to have a good defensive weapon, and venom does that trick. And in fact, Justin Smith has made an entire career out of it, basically driven by the question, which venoms are most painful? Because pain is the currency of defensive venom. Immediate, memorable pain is what these creatures use to defend themselves against mostly vertebrate predators. So he asked questions, are socially stinging insects more painful than solitary ones? He has allowed himself to be stung hundreds of times. Not always voluntarily, I have to say, but he made a scale from 1 to 4 about how painful it is. 1 is like, meh. 4 is lie on the floor and scream. And he has tasting notes for this. I would say consult this book called Sting of the Wild. It's a wonderful read about what not to do when you're in the forest. Give you just one tasting note. If you get stung by this warrior wasp, he knows it's like torture. You've been changed into an active volcano. Why did I start this list? This is suffering for science. It's 25 minutes. Can I go a minute and a half over? I can. Some experiments are weird. I mean, you can experiment like Justin did. This is a assassin bug, for instance. An assassin bug killing a scorpion. This is a creature with the proboscis. It injects venom, it paralyzes the prey, then liquefies it completely because it's extremely strong dissolving venom, and then slurps up the soup through this proboscis. But why would you do this? Why would you take one of these, and why would you allow it to sting a mouse in the lab and see it dies within 15 seconds? Why would you aerosolize the venom to see how long it takes to kill a guinea pig? Why would you do that? I don't know, actually. I don't have an answer. This is from the same issue of that journal from the box Jellyfish experiments. If you have a real nasty Halloween spirit, then tonight I would go online and read this journal. No, you shouldn't do it. It's bad. Now we have far stricter regulations for animal experiments. I hasten to add. Last example. Why would you do experiments with, say, rats and mice and a Brazilian wandering spider? They bite hundreds of people a year. Some die, many get horrible symptoms. Well, there's a reason for it. If you want to turn the bad power of venom into good, you need to isolate it into its components so you can understand them individually, and then you can perhaps make something good out of it. This is why people inject components of the spider venom into mice and rats and notice that you get priapism in death. This is where the genital bit of the story is. What does that mean? Well, you inject this venom component, and then you read the abstract, and they say this. The most remarkable histological finding was a general vascular congestion. Basically, the blood vascular system was destroyed. But, outstanding result, the penis had no necrosis or damage. This, for a venom researcher, is an encouraging result, because if you look in, this is actually a little boy, and has been bitten by one of these, and this is a horrible picture I don't have to point out, and this is a rat with the same thing. If you use this peptide, you can reliably induce erections in diabetic rats, fat rats, hypertensive rats, and old rats. These are the risk groups for erectile dysfunction in humans. A quarter of the people, of males younger than 69, have erectile dysfunction, a much higher percentage in males older than that. A third of those don't respond to things like Viagra. This is a problem. So if you find something that can act on that system and create an erection reliably, you might have a new drug. There is a problem. The narrow range of concentration in which it gives you an erection before it literally kills you. But because if you give these rats a little bit too much, they get an erection first, and then they shiver and paralyze and die. But they now have developed a synthetic peptide. They tweaked it, that all these side effects are gone. And now they can start clinical trials on humans. This is hugely encouraging for people with problems like this. And this is already delivered blockbuster drugs. This is a Gila monster. And a Gila monster, I will literally be done in a minute and a half. A Gila monster has a venom used for defense, and one of the components regulates glucose levels in the blood. And it's already been turned into a type 2 adult diabetes drug that is worth $800 million of sales a year for AstraZeneca, a pharmaceutical company. Helps thousands of people a year. They no longer need insulin. If you use this variety of the treatment, you only have to inject yourself twice a week rather than after every meal. And so if you look in the venoms of different animals, as different as snakes, leeches, cone snails, and sea anemones, you can discover that you can do good stuff with venoms. Just as an indication, the sea anemone venom is paralytic, and one of the toxins that paralyzes crustaceans, that's what it does for the animal, is used successfully to treat autoimmune diseases like psoriasis, and they're trying to tweak it now for multiple sclerosis. You can help thousands, if not millions of people better their lives. Lunch is coming. I've talked about bad stuff, I've talked about good stuff. Is venom everywhere, even in the UK? Yes, we have about 8,000 venomous species. Most of them are tiny parasitoids, so don't start screaming yet. What if you are lucky enough to eat this for lunch? You have some octopus, some melon, and some figs, with some reduced red wine sauce and some lemon juice. How many venomous animals are needed to make this dish? Well, at least 5. Well, you need to have bees who have defensive venom to pollinate melons and other plants. The octopus itself is as venomous to crustaceans as snakes are to us. They paralyze them, and their venom then dissolves their muscle from the exoskeleton so it can then slurp the spaghetti out. If you want to grow citrus fruit like lemons, you need these little parasitoids to make sure that the pests don't eat your trees. Figs need parasitoid fig wasps that use their venom to manipulate the plant to create a breeding chamber for themselves, but without the fig wasps, the figs themselves cannot reproduce. And then finally, this is a sea catfish. They commercially harvest the swim bladders of these and process them so they can clarify red wine with them. So you have to be careful if you're a vegetarian or vegan with anything anyway, but sometimes you actually ingest probably remnants of venomous fish bladders as well. Now, the very last slide, if this whetted your appetite, as I hope it did, last year we published a lovely little book, not surprisingly titled Venom. This is my friend and colleague Ivan Undheim from the University of Queensland. You may or may not want to read this. It's richly illustrated. If you really like a challenge, it's available in Dutch. And if you really like a challenge, since Friday, you can get it in Japanese also. But with this, I will stop talking and thank you for your attention.